Hello, everyone, and welcome to today's live broadcast, Liposomes for Drug Delivery and Targeting, Examples of Their Potential from Our Laboratories Research, presented by Sophia Antimisiaris, Professor, University of Patras and Institute of Chemical Engineering, 4th ICE HT. I am Alexis Corrales of Laboratories, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Laboratories. Laboratories is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Accreditation button located in the promotional board at the bottom center of your screen and follow the process to obtain your credits. Please join me in welcoming Dr. Antimisiaris, and I will now turn the presentation over to her.
The presentation, as you already heard, the title is Liposomes for Drug Delivery and Targeting, Examples of Their Potential from Our Research Results. And you already heard where I am from. Just a bit of an introduction about the University in Patras and the Institute of Chemical Engineering, which is part of the 4th Institute, the largest institute in Greece. We are located in Patras, which, as you see in the map, is in the central part of Greece, on the side towards Italy. And these are a few pictures from the area where our university and our institute is located. The outline of my talk is seen over here. Initially, I will talk about the advantages of nanotechnologies in general compared to conventional formulations. What is the advantage that we get if we administer drugs in the form of nanoparticles or nanomedicines, as we may call them, compared to the usual formulations that we know. Then we will focus on liposomes and talk about potential applications with examples from the work that we have been doing in our laboratory in the last 20 maybe years. So let's go on with the first part about the advantages of nanotechnologies compared to conventional formulations. When we administer conventional formulations, we usually get fluctuations in the concentration of drug in the serum and in the organs where we want the drugs to show their pharmacological action. So this may not be very good for the therapeutic effect. And in many instances, we want to control the release from the formulation in order to have better results. Also, when we administer conventional drugs, what we get is that the drug, after it is absorbed in the blood, distributes to all the parts in the body, depending on the physicochemical properties and the size of the molecule. And this means that it will end up eventually in the site of action, but also in other tissues where it may cause toxicity and side effects. So if we somehow could administer the drug in the form of nanotechnologies, we can have several advantages. As you see, they may be protection of sensitive molecules from areas in the body where the molecule may be destroyed, control release, and also targeting to the specific site that we want the drug to go to. And this way we will have increased pharmacological activity and decreased toxicity. Just a brief introduction about what nanotechnologies in general are. They are nanoparticles, usually in the form of spheres or capsules. Their size is about 10 to 200 nanometers. If they are round, this is the diameter. And they can be loaded with active drugs. They can control the release rate of the drug, and they can transfer the drug to specific targets in the body, assuming that they have some targeting moiety on their surface. There are different types of nanoparticles. One of these types is the liposomes, and the structure and the ingredients may vary. They may be polymeric, they may be lipids, they may have a structure of capsules or spheres, they may have other structures such as dendrimers or micelles, and they may also be composed of inorganic materials. But of course, in this case, there are several problems when we want to apply them for delivery of drugs. So what are the requirements that we need our nanotechnologies to have in order to be used for drug delivery? As you can see here, it should be possible that we can load them with adequate amount of active drug. As higher the loading, the less the amount of nanoparticles that we have to use. So this also may have an effect on the toxicity of the particle that we use. It has to be easy to modify the nanocarrier surface, because the surface is very important for the actual result, because this is what the body sees. They have to be non-toxic, non-immunogenic, biodegradable, and biocompatible. And all of these, of course, depend very much on the dosage we administer. So if we can have high loading of the drug, we can have high drug concentrations with a lower amount of nano material, and this means that we can have a lower dose and minimize the chances of toxicity and problems with biocompatibility. Also, we have to be sure that the drug is retained in the nanoparticle until the biological target is reached, and we have to be sure that it will be actually released when it reaches the site where it has to act. So all of these are important, and as I already pointed out, loading and dose are two characteristics that are very important and determine, let's say, the possibility to use any type of nanotechnology for drug delivery. And now we will go to the second part, because we will focus on liposomes. In the last 20 and so years, we are working in our lab with liposomes. So I will try to convince you that this is the best system to use when you want to deliver drugs. And then I will show you a few examples from our team's research about their potential applications. So first of all, let's see what liposomes are, and let's talk about their advantages and potential disadvantages. Now, as you can see in the cartoon on the top of this slide, liposomes are actually composed of lipids, of a lipid bilayer. It's something like a cell membrane. The lipids are phospholipids, and they have an aqueous entrapped center where we can load hydrophilic drugs or other type of particles if we need to. So the first advantage is that we can have high loading efficiency, and we can load any type of drug, because we can have lipophilic drugs in the membrane or hydrophilic drugs in the aqueous center. Another very important parameter and advantage is that they are non-toxic, fully biodegradable and biocompatible, non-immunogenic, because they are actually composed of phospholipids and cholesterol maybe, which are the main ingredients of our cells. And they are very versatile, so we can easily change the surface as we want to. Perhaps we want to add something on the surface that will direct the liposome to the specific part in the body. So it's easy to do this with liposomes because of the versatility of the chemistry of the lipids. Also, they have ability to modulate the release of the active molecules by different triggers if we want to, or by simply modulating the lipid membrane composition. And I will show you some examples further down. One disadvantage, if we want to really be strict, is that the amount of time that a drug can be retained in liposomes may be shorter than the time that we have in other types of nanotechnology, like polymeric nanotechnologies. Nevertheless, these disadvantages can be overcome by construction of hybrid systems, and I will show you a few examples further down in this talk. So going on, I have to point out that when we talk about liposomes, we can categorize them in 4 different main classes corresponding to their ability to transfer drugs. So we may have conventional liposomes, which are liposomes composed of phospholipids with nothing on their surface. The problem with these types of liposomes is that they end up very fast in the liver and spleen after we inject in the body. And this is because they are taken up by the RES of the immunological system, the macrophages in general. And this way, the potential to use this type of liposomes for drug delivery is very limited because we can only reach the macrophages. And this is called passive targeting in this case. If we want to reach other sites in the body, we have to have liposomes that circulate for longer in our blood. And in order to do that, we have to have on their surface specific type of molecules like polymers or sugars that will make the surface hydrophilic, and this way the liposomes will avoid to be taken up by macrophages and will circulate for longer in the body. And this gives them chances to reach other sites as cancer tumors, for instance. The third type is the targeted liposomes. In this case, we want to have a targeting ligand that will target specifically our liposome to a specific cell type in the body. And the last type is the cationic liposomes, which are usually used when we want to deliver genetic material, because there is a possibility to connect the genetic material with the cationic lipids. I will not be talking about cationic liposomes and gene delivery today because the examples I will show you are more general examples for drug delivery, conventional drug delivery, let's say. Okay, so let's go on to the next slide. And now I'm going to go into the third part about the potential applications of liposomes. I will show you how we can use liposomes to simply protect active molecules from areas in the body where they may be degraded, so inactivated in other words, or vice versa. We may want to protect the body, the cells, from drugs that are cytotoxic at high concentration. So I will show you some examples about this first of all. Now, to protect the body from very active drugs or toxic drugs, we may use this type of nanotechnologies. We may use liposomes or other type of nanoparticles. This is an example about the reduction of cytotoxicity of rifamycin. Rifamycin is quite a toxic drug, so when it reaches alveolar cells, it has some cytotoxicity. This has been done in my lab some years ago, and we can see in the graphs on the left side that the viability of the cells is lowering as we increase the concentration of the drug. The columns that are white are free drugs, correspond to the free drug, while the other columns correspond to different types of nanoparticles composed of polymers in the top graph, and liposomes, different types of liposomes in the bottom graph. So we can see that as we increase the concentration of the drug, we have more cytotoxicity when it's in free form, but if we have it encapsulated in polymeric particles or in liposomes, we can have a very high reduction of the cytotoxicity. So we can administer high concentration of drug without having the toxicity. Now, a second example shows us the opposite, how we can protect a specific drug from the environment that it goes into when we administer in vivo. In this case, we are talking about, we are showing the example of curcumin. Curcumin is degraded in aqueous solutions very fast. You can see in these two graphs, the black line, the bottom line with the squares, is free curcumin when we put it in serum at two different concentrations, 0.01 milligram per ml and 0.05 on the right. So we can see how fast curcumin degrades. When we add curcumin in different types of liposomes, we can protect it. And we see that we can prepare liposomes that protect the drug, and this way we can administer the drug and have it in an active form for longer periods of time. So we expect also a higher pharmacological action. Now, the second thing I'm going to show you examples about is how we can control the release rate of drugs when we administer in the form of liposomes. I already pointed out in the beginning that maybe a disadvantage of liposomes is that they cannot retain drugs for very long periods of time. This, of course, depends on the drug properties. I will show you, to start out, this example where I'm showing you here on the left side the release of heparin from liposomes, two different types of liposomes. So we can see that we can change, especially for such large hydrophilic molecules, we can control their release very, very nicely, and we can change the release kinetics by using different types of liposomes. Actually, what we are doing is we are using different lipid membrane compositions. So we have, in the one case, non-saturated lipids, and in the other case, saturated lipids, and we can see how we can control very nicely the release, and for several days over here, have very low release, or faster if we want to. Now, if we have a smaller molecule like calcein, it's hydrophilic, and you can see these results on the left side, on the right side. You see that it is released faster because the time is hours now and not in days. And again, we can control the release depending on the liposome composition that we use. And in the middle graphs, you see the retention. Actually, this is not the release. It's the retention of dexamethasone, which is a more lipophilic drug. So it is released also pretty fast from the liposomes. But again, we can use different types of liposomes and control the release. Of course, we cannot control the release for months. And in this case, if we want to have a longer release, we can use different types of hybrid systems. So I will show you three types of hybrid systems. The one is the drug in cyclodextrin and then in liposome system. The other is to put our liposomal drugs in gels. And the third one is to actually mobilize our liposomes on surfaces of biomaterials in order to produce drug-eluting biomaterials. In this way, we can also control the specific area in the body where our drug is released. And we can have a different mode of release depending on the liposomes that we use. Okay, let me go and show you now specific examples for each one of these cases. Let's go to the first one. Why would we need to have the drug in cyclodextrin in liposome system? To make a long story short, in some cases when our drug is lipophilic or amphiphilic, it actually has a possibility, it has the capability to permeate the liposomal membrane. So if it's somehow partitioned between the lipid and the aqueous phase, the time that we inject it or we administer to the body and it reaches the blood, because of the dilution, it is very diluted, so some of the drug is released from the liposome and saturates the extra solution that is available, the extra water that is available in the system. So we very fast, very rapidly lose a lot of the encapsulated molecules of drugs. So in this case, what we can do, and this was proposed several years ago by the group of Gregoriadis, we can actually have a complex of our drug with a cyclodextrin molecule. This complex cyclodextrin are molecules that have an interior. They form like a capsule with a lipophilic interior and a hydrophilic surface. So our lipophilic drug will go in the capsule. It will form a complex, and the exterior of the cavity, of the cyclodextrin cavity, is hydrophilic. So this way we can increase the solubility of a drug and actually encapsulate it in the center, in the aqueous phase of the liposomes, and this way it is more stably encapsulated in the liposomes. We have done this work in our laboratory with prednisolone and with curcumin, and in the publications that appear on the end of these slides, you can see, if you want to, you can go to these publications and see what results we can have compared to the plain liposomes, let's say, when the drug is just partitioned between the aqueous and the membrane phase. The second case I was going to talk about is about drug in liposomes in gels. So if we have a small drug in a gel system, it may be released very fast. One way to decrease the release rate is to encapsulate the drug first in the liposomes, as you see in the body, and then in the bottom cartoon, and then in a gel. This way we have two diffusion rates, one of the drug to release from the liposome and then from the gel, and this way actually we have a slower release. So we can control the release from such lipophilic gels and have a more prolonged retention of the drug in our gels when the drug is initially entrapped in the liposome, and then this liposomal drug is dispersed in the gel. So here you see what we have. This was work that we did in the content of a European project some years ago. And on the left-hand side, we have the plain gels with the drug inside. And on the right-hand side, we have the liposome gel. So we see, if we put them together, that, okay, the release is prolonged when we have the liposomal gel. And on the bottom, you can see that we can control the release rate by feeding our system with different amount of liposomes. So it's very easy to work this way and have a more prolonged release from the liposomal gel compared to the control gel. This is one example. We have a second example here where we can control actually the release of drugs from liposomes by incorporating these liposomal drugs in gels that are also pH sensitive in this case. So this is work that we published in 2011. You can see the reference in the bottom of the slide, and you can see in the right hand side there is a graph where it shows the release of the drug from plain liposomes or the release of the drug when we have the liposomes incorporated in this polymer. We can adjust the weight of the polymer, the concentration of the polymer, and have a different release rate. And actually we have a zero order release, which is very important to get constant concentration in the blood. So this is a very nice example of what we can do with such lipophilic gels. And let's move on and see another case from a very recent work that we have been doing in collaboration with a group from the University of Crete. What we wanted to do is to sustain the release of a small non-steroidal anti-inflammatory drug, flurbiprofen, after intravitreal injection in the eye. So we can see that the drug is very fastly released from the gel when it's free. It's very, the black line shows us how it can be retained when we add it in a polymer. And when we make a hybrid system with the liposomes in the polymer, we have the red, where we see that we can control the release and have a sustained release for a very long period of time. The plain liposomes do a good job, but not so good, and that is the green line in our graph. So we can see that we can increase the mean residence time. On the bottom, you can see the pharmacokinetic parameters and also the AUC that we have in the vitreous. And of course, we also have very good results in the retina. And if one wants to see more about this, you can see it in the reference that I quote over here, a work that we just published last year. So going on, the third, let's say, hybrid liposome system that I talked about is the drug-eluting biomaterials. We have done several different cases. In one case, we immobilized liposomes on polymeric materials from polymer-coated stents. And you see in the picture on the left-hand side, the liposomes attached, actually absorbed on the polymer. They look like lines, okay? So you can see the liposomes in there. And we can control the release of the drug by using different type of liposomes. And as you can see in the graph on the top. And this was done just by simple absorption of the liposomes on the material of the stent, on the polymer grafted stent. But we also moved on and we found out that we can immobilize liposomes on plain metal surfaces after we graft specific functional groups on the surfaces with plasma technology. So these plasma-treated surfaces were possible to interact with functionalized liposomes that we prepared. And in the liposomes, we had put heparin, and this resulted in a very significant increase of the biocompatibility of the material. You can see the biocompatibility graph on the bottom, where the blue columns show us the case where we have the liposomes attached covalently on the surface of the materials, and having heparin inside, the heparin is released, and therefore the biocompatibility, the hemocompatibility of this material is very much increased. We also did the same similar thing to produce antimicrobial surfaces by using topramycin liposomes, which were immobilized on the surface of catheters. So you can see here in both graphs, the case where the columns are very low are actually the cases where the catheters were coated with these liposomes, which had topramycin inside, so bacteria could not approach on these surfaces. Actually, what we measure here is the amount of bacteria that are forming on the surface of these catheters. And we all know that this is a problem. So this is an example of what liposomes can do to produce antimicrobial surfaces. And this again was published in 2015. Now let's move on to the last part. The last part I think is the most important and the most challenging. And it's actually about targeting. Okay, what can we target with liposomes? We can target more or less everything, but I will show you now some examples from our work about targeting cancer cells or cancer tumors, biological barriers, such as the blood-brain barrier, which is the most challenging and most important barrier in our body, I think. And we can also target specific pathologies related with diseases, such as the amyloid plaques in Alzheimer's disease. So I will show you some examples from work that we have done years ago or recently. First of all, let's talk a little bit about cancer. I will go very fast. If you don't know a lot about this, I think you would have to find a reference and understand more about it. But to make it very short, in cancer, there is a phenomenon that is called EPR, for enhanced permeability and retention effect. And this means that because of the very fast angiogenesis in the cancer cells, the vasculature that is formed is very leaky. So if we have liposomes or any type of nanoparticles that are very small and circulate in our blood for long periods of time, eventually they will extravasate in a tumor, if there is a tumor around, because of the leaky vasculature. So because of also the very low lymphatic system, they are actually trapped there, so they can release the drug. This is why we have very good performance of liposomal drugs for several types of cancer. But we have to be sure that we are preparing our liposomes in the correct way. First of all, we have to have a very good polymeric coating on the surface, so they will be able to circulate for a long period of time, or be, in other words, we call this stealth. So they have to be very stealth. And also, we have to be sure that if we are adding a targeting ligand to specifically target a specific component on the surface of the cells, like a protein which is overexpressed there, we have to be sure that the ligand that we add is on the surface of the liposome and it's not blocked up by the polymer coating. So it's important how we prepare our liposomes. Here I show an example of targeting HepG2 cells in the liver by using liposomes that have on their surface an acylfetoine targeting ligand. This ligand will target the galactose receptors, which are overexpressed on these cancer cells. So as we increase the distance between the ligand and the surface of our liposomes, as we go from the top to the bottom in these graphs here, we see that the targeting we get in the cells compared to the control cells, which are the black symbols, the white ones are the amount of liposomes that we get in the target cells, we increase the targeting actually. So when we prepare our liposomes in a correct way, in this in vitro setup, we saw a very good effect. Of course, when we go in vivo, the results are not so good, and this is why we don't have today anything on the market which is targeted. This is something very important at the time. Now, going to the blood-brain barrier, we have done a lot of work through the last years, and this was within an FP7 European project. It was funded by Europe. It was in collaboration with many other labs, and we have published a lot about this, and you can see the publications over here. So what we have done is we prepared liposomes using one ligand to target one receptor on the blood-brain barrier and another ligand to target another receptor. And when we have both ligands on the same liposomes, we see on the top graph, we see the uptake by specific blood-brain barrier endothelial cells, and we see that the black one is the control liposomes with nothing on their surface, simply pegylated liposomes with a polymer that will make them circulate for long. And as we add one ligand or another ligand, we have an increase compared to the non-ligand liposomes. And when we add both, this is the green column, we see that we have a better effect compared to the liposomes that have one ligand on their surface. And we have the same results. We had the same results when we checked this, how these liposomes transport across a cell monolayer cellular model of a blood-brain barrier. So if you want more details, you can find them in these publications, but let me go on saying that we also did a lot of in vivo experiments with this system, and we saw the same effects. If we have one ligand, we have a little bit increase. If we have the other ligand, we have again a similar increase. But if we have both ligands, the red line in the graph on the right hand side, we see that we have a best results, and we see this actually in the brain of the animals. The last animal, we see the signal in the head is very much increased because it has it is the liposomes with both of the ligands on the surface. In this, after we finish this work, we are continuing our work now, trying to increase the potential for targeting the brain with liposomes by using, by getting some influence from the organotropic properties of extracellular vesicles or exosomes. This is work that we are doing now, and we hope to have even better results from the ones that we achieved over here. Now, let's go on and talk about the third part of targeting, actually, how we can target specific pathological features. And in this case, what we wanted to target was the amyloid plaques, or actually the ingredients of the amyloid plaques, which are amyloid peptides in the brain, in order to find a solution or a therapeutic or diagnostic system for Alzheimer's disease. And again, this research was funded by the same project that I talked about before. So what we tried to do here is find specific ligands that we can put on the surface of our liposomes in order to target these amyloid peptides or amyloid species in general. We use different types of curcumin because from the literature it was known that curcumin can have some affinity for beta peptides. So we synthesized two types of curcumin derivatives, and we immobilized them on the surface of liposomes, and we found out actually that these liposomes have very high affinity for different types of amyloid species. So they were also able to have an effect on the aggregation of amyloid peptides. Amyloid peptides aggregate, they form the plaques, and the plaques are responsible, or the oligomers of the plaques are responsible for the toxicity that is demonstrated and the pathology that is demonstrated in Alzheimer's disease. So these liposomes were actually able to decrease the rate of aggregation of amyloid peptides, and they were also able to, as we see in the right-hand side in the top, to stain amyloid plaques from brain slices post-mortem of Alzheimer's disease patients. And they also had the possibility to lower the toxicity of beta peptides towards specific types of neural cells. So we thought that these liposomes could be able to target Alzheimer's disease And we, at the end, what we did is that we prepared liposomes that are multifunctional in terms that they have ligands to target both the brain, because they have to pass the brain to get the blood-brain barrier to get into the brain, and also the amyloid plaques. So when we added all of these ligands on the liposome surface, we see on the cartoon on the left side, we have 3 ligands now. In fact, we didn't see any difference in their ability to target the brain. On the bottom graph, we see this result from our in vitro cellular system, the monolayer system. The transport is the same for the liposomes that have only the blood-brain barrier targeting ligands and the ones that have also the ligand to target the amyloid plaques. And when we administered these in wild-type mice and in Alzheimer's disease model mice, double transgenic mice in this case, we see that the amount of the liposomes that end up in the brain are much higher in the transgenic animals in the Alzheimer's disease model compared, this is a red line, okay, compared to the wild type animals that don't have amyloid plaques in their brain. So perhaps this is because of the amyloid plaques. Of course, we need to do more work to prove this at the end. So, but this is a very good result for the time. So I hope from here that we can conclude, we can come to the conclusion part of this presentation. I hope that I convinced you about the advantages of nanotechnologies compared to conventional formulations. I hope I convinced you about liposomes and what they can do, the limitations to control drug release and how we can fix it with hybrid systems, and also how we can use liposomes for specific applications when we want to increase the protection of drug molecules. This is a simple situation. When we want decreased cytotoxicity of large concentration of drugs, or when we want to increase the targeting of specific cell types, cancer cells, biological barriers, or pathological sites of, or molecules like the amyloid species. And here I have to come to the acknowledgement of all the contributors in this work that I showed today. Most of the people are from my lab. I have here very many previous and recent researchers working in my lab. Also from the University of Patras, there are other colleagues that I collaborate with, like Professor Klepetsanis, Professor Stathopoulos, Professor Misilis and Tsitsilianis, and also from the University of Crete, Professor Tsilibaris and his group, and several groups from abroad that contributed in some of the research that I showed today, in some of the results that I showed today. So you all see them here. And I want to thank you for your attention.
Thank you, Dr. Antimisiaris, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, which routes of administration can be used for liposome drug formulations?
Yeah, we can use different routes of administration. But of course, you will see in the literature, mostly liposomes are administered IV, intravenously. But they can work very well also for topical administration or for local administration. There are different types of liposomes that can be used for transdermal delivery of drugs. I didn't get into that in my presentation, although we have worked on it. So the only problem with the liposomes is that you cannot have very stable liposomes in the GI tract because of the cholic acids in the GI tract. So that's the only route that is not very possible for liposomes.
Our next question is, are there liposome formulations in clinical use as drug products today? Which drugs are currently available in liposome form in the market?
Yeah, there are many drugs in the market. The first ones that appeared and are already in generic formulations are doxorubicin, which is an anti-cancer drug, and amphotericin B. These two drugs have very good results, and mostly because with the liposome formulation, the pharmacokinetics is changed. So this means that the drug eventually ends up in different areas in the body, and we have very significant decrease in their toxicity. These drugs are very toxic. So this way we can increase the dose without having the toxicity, and this means that we have a better pharmacological action and a very good result. So there are several drugs on the market today, and FDA has approved for several, mostly for several types of cancer.
Which is the advantage of the current liposome drug products, and what is the theoretical background that explains their advantages compared to the corresponding conventional formulations?
This is connected with the previous, what I said before. These formulations that are on the market today mostly work because they decrease the toxicity of the drug because of the change, the modulation of the pharmacokinetics compared to the pharmacokinetics of the free drug. And this is the theoretical basis. So today we don't have any targeted drug formulation, liposomal drug formulation on the market, but mostly passively targeted formulations. And this is why most of the formulations are for cancer. I already explained about the EPR effect in my presentation, so this is actually the theory behind most of the existing liposomal drugs on the market.
What are the current challenges in the field of nanomedicines for drug delivery?
Current challenges are related to this, that we don't have a targeted formulation of any drug with liposomes on the market today. Although there has been a lot of research during the last 25 or 30 years with very good in vitro, in some cases also in vivo results in animal studies, there is no translation to a market, a product for clinical use. Mainly because the difference that we have between the liposomes and the targeted liposomes is not yet so good to advance to this type of formulation. The other big challenge is, of course, to be able to cross biological barriers. And the most important, of course, is the brain, the blood-brain barrier. So these two challenges are very, very important. And I would add also another challenge would be to have a formulation that can actually deliver oligonucleotides in order to get the advantages of this type of therapeutics in the clinic.
And it looks like we have time for one more question. Do such nanotechnologies have applications in other fields, such as cosmeceuticals, food products, etc.?
Of course, of course. There are many applications of liposomal formulations, let's say liposomal encapsulated enzymes or liposome encapsulate active materials that could be also integrated in cosmeceuticals or food products. Ripening of cheese is something that can be done with liposomal enzymes. There are several examples of the use of liposomes in non-medical, let's say, fields. So it's an attractive methodology to use also for other types of, in other areas where you want to control the release of a specific agent from a specific formulation.
I would like to once again thank you, Dr. Antimisiaris, for her presentation. I would also like to thank Laboratories for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from Laboratories letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.